Hello, everyone. Welcome to, I think this is our sixth or seventh year of doing the Regional Soybean Gall Midge webinar. We appreciate everyone's time. Yes, welcome. I think there's going to be a few more people joining us, but first of all, we just like to thank, we have a lot of sponsors and a lot of supporters. And some of that comes from commodity checkoff dollars, especially the North Central Soybean Research Program and the United Soybean Board, and then presentations and people power that we're going to highlight today. Also, it comes from the Iowa Soybean Association, the Nebraska checkoff, and the Minnesota checkoff. We also get some different types of funding from federal resources like the North Central IPM Center and the USDA NEFA program, so we appreciate that. And then we just have a lot of people involved with extension from our representative states, including Nebraska, Minnesota, and Iowa today. So thank you for those. We couldn't do it without you. But we'd also like to thank Phyllis Bongard and the University of Minnesota Extension Group. They help with registration today. They asked you a couple of questions, and some of you did that, and we thank you. And also just managing the tech behind the scenes with our webinar. So thank you, Phyllis. I did mention this is our sixth year, but if you want to watch any of the previous webinars, if you'd like to see more information about the distribution, sort of real time as it might happen this year, we encourage you to visit just our website that we push all the information to, soybeangallmidge.org. So don't want to forget about that. This is a brief agenda of the topics that we're going to cover today, and they're going to be sort of like speed dating style. So we're going to go quickly through these. We hear feedback that most of the audience, that they appreciate shorter presentations instead of longer ones. So we're going to give a lot of short updates today on the research that's been going on in the last year, maybe a couple years, depending on the topic. And we'll try and answer those questions that you sent ahead throughout, but then we'll just leave some open time at the end to try and cover all the things to the best that we can at the very end of the webinar. So one of the first questions that we get when we talk about soybean gall midge at extension and education events is, where are we with the distribution map? So I thought I would just start with that before we got into our topics. And thanks to Justin, we have an updated map from 2025. We have monitored the first detections in counties throughout the United States for 8 years. We started in 2008. You can see all those red counties that showed up. And we've been monitoring ever since. Right now, we are at 7 total states that have at least one county infested with soybean gall midge, and we're at 185 total counties. I think this represents just over 2 million total possible acres of soybean that could be infested every year. Maybe that's not quite right. It's probably more than 2 million actually, but we had, as of last year in 2025, 5 new counties in our region, 3 of them in Iowa, and then we had Minnesota, and then also Nebraska. And then you can see a couple of shaded counties that have like light and dark stripes, and those are counties that we can find just the word larvae were just found on clover, sweet clover. And so Justin already corrected me. It's over 19 million. Now, when I said 2, I was like, that's definitely not right, because that's there's more than that in just Iowa. I don't know where I got that number. So at least 19 million acres are impacted, potentially impacted by this pest. So we always get lots of questions about the map, so I thought I would just start with that. We are going to move on to our first speaker. We are going to start with some of the 2025 interactions and impacts from our first speaker. He's a student at the University of Nebraska-Lincoln, Talis Rodriguez da Silva. Talis, are you able to share your slides? I see them coming up. Great.
All right. Thanks for the introduction, Dr. Hodgson. My name is Talis. I'm a master's student in the University of Nebraska-Lincoln. And today I'm going to share with you some of the results of my research on soybean gall midge, dactys, and their co-occurrence on soybeans. So moving to our introduction, a brief introduction to Soybean Gall Midge. So early in the season, just let me set my pointer. Perfect. So early in the season, we have the emergence of midges here in Nebraska around late May, June. These midges, they will look for fissures in the lower stem of the soybean plant. They're gonna lay their eggs here. Their larvae will start feeding on the tissue, usually also including the xylem and the phloem. And by feeding on those tissues, they destroy that and they block the flow of water and nutrients from roots to above ground plant parts, leading the soybeans to wilt and die. Moving to our next pest, Dactyes, they we also start the season with the emergence of overwintering beetles. They will attack the upper third part of the plant. So here you can see a female beetle chewing the petiole. Here you can see this puncture. So they're going to lay an egg in the petiole. The larva will initially feed on the insides of the petiole. As the larva grows up, they move to the stem where they're going to feed up and down, also in the stem insides. And later in the season, when plants reach maturity, they move to the lower stem where they will girdle the plant, leaving the plant with a fragile structure and more likely to lodge. Now talking about the co-occurrence, the co-infestation of this pest, here we have a great picture demonstrating that. So this is a petiole, and here you can see the puncture made by Dactyes, which was potentially used by soybean gall midge later eggs. And you see that this petiole also doesn't look healthy. It's slightly darkened, discolored. And when we split it open, we found gall midge in an unusual part of the plant. And we also found gall midge in the stem, usually on the galleries, where we were supposedly to find Dactyes. And here you can see where the Dactyes moved from the petiole to the stem, and this breach was potentially used by gall midge for invading this tissue of the plant. So here we have a co-infestation time frame. You can see that gall midge starts at V2 and Dactyes comes a little bit later on V5. But the highlight of this slide is that from V5 to R7, you can have co-infestation of both pests in regions where they co-exist. So Dactyes is here in Nebraska, just like gall midge. So we have a long time for both pests to co-infest the same plant. Moving to methods, my study was conducted in 9 sites across eastern and south central Nebraska. We had 5 treatments. I'll start with untreated, where we just planted the soybeans and harvested. We had no intervention. I also had a disease management treatment. However, I won't talk about the results from this treatment because that's not the focus of this webinar. For controlling Dactyes, we had fipronil seed treatment. And here in this picture, you have a Dactyes larva that died feeding on a fipronil treated plant. And for controlling gall midge, we use healing. So we pretty much in between rows, we use a tiller to soften the soil, and we push the soil to the base of the plant, covering those fissures and blocking the midges to infest the plants. And our last treatment was the combination of fipronil and healing to control both pests. This study was conducted in a randomized complete block design. We conducted the study in the field edge because that's in that spot is where we have the greatest source of gall midge and Dactyes. And now moving to results. So starting with soybean gall midge with larval abundance, I highlighted here in red four sites on southeast Nebraska where we had the highest average soybean gall midge larvae per plant, ranging from 8 to 18. And when we compare the untreated larval counts to healing larval counts, we see a 95% soybean gall midge reduction. So our treatment was effective in controlling gall midge. Moving to yield, we can see that on those same sites where we saw greater gall midge larval abundance, we also noticed high yield loss ranging from 17 to 38 bushels per acre. And when we compare the yields from healing against untreated over these 9 sites, we see that gall midge potentially caused a 24% yield reduction. Moving to our second pass, that stem borer, we see that from those 4 sites that we had high gall midge, we have 3 here and another site on east. And when we compare fipronil treatment plots versus untreated, we see a 66% in that stem borer larval abundance reduction. And now moving to yield, we see overall we don't see large losses overall. We had 2 sites with that had a 13 bushels per acre loss. However, when we look the big picture, we see only a 4% yield reduction comparing the fipronil treated plants versus the no treatment plants. Now moving to co-infestation, we see that those same sites where we had more dacties, we also had a greater co-infestation of gall midge and dacties in the upper part of the plant. And when we check the average number of soybean gall midge larvae in those co-infestations, we don't see numbers as high as we see in the bottom part of the plant. So we saw an averages from 2 to 5.5 larvae in these co-infestations. And now showing the yield impact overall. So when we compare healing with controlled gall midge versus fipronil, where we controlled dacties, we see only a 20 not only, it's a huge loss, 20% yield loss. However, when we compare healing and fipronil plus healing, we don't see a huge difference. So this highlights the impact, how impactful gall midge is, and dacties, it's not that impactful on yield. So the summary of this presentation is that heal and fipronil were effective treatments to control gall midge and dacties. On sites where we found more gall midge in the plants, we also found greater losses. Dacties was a potential threat to yield in 2 out of 9 sites, but overall was only a 4% yield reduction, so not a big deal based on these 9 sites. In the prior slide, we can clearly see that gall midge was the main driver of yield loss, and we also can see in the map that southeast Nebraska was the main spot, was the hot spot for both pests. So I'd like to thank all my lab members for helping me with this big study. And I'd also like to thank Nebraska Soybean Board for funding and the University of Nebraska-Lincoln for the support. So if you have any questions, I'll do my best to answer them. Thank you.
I think that most people realize that fipronil is not labeled as an option for soybeans. So I'm curious to know why you tried it, maybe in some of the different treatment combinations that you showed us today.
So, oh yeah, sorry, I forgot to say this in my presentation. So we use unconventional tactics. So healing is not common for soybeans. It's more common for potatoes. And fipronil is not, like you said, it's not labeled for soybeans. So the goal with these treatments is to have a, I want to say, is to remove one of the insects of the equation. Not remove, but I mean keep them really low, so we could assess the impact. So where we have like I think this uh, my last result slide really shows that. So for fipronil, we remove Dactyls and we checked their impact when we compared to untreated. Oh, then we remove Gall Midge and we compared to check. So we use these treatments to control the pests and make a comparison to the other treatments so we could evaluate their impact on the soybean yield.
Mhm.
Did I
Yeah,
answer your question?
yeah, I think so. And then, as you mentioned, it's the healing does seem to be really effective as far as deterring or limiting infestations, but it's probably not something that's really common to most people, as you said, it's used for potatoes. So can you describe a little bit more how you're moving that soil to the base of stems?
Perfect. Let me go back on my method slide. Yeah, here, so we have a healer, Justin operates that. So it's there's a machine for that can heal the plants and it works great. Yeah, so on this study, repeating that's not feasible. We use a tiller in between rows and then we soften the soil, then we push the soil to the bottom of the plant. So we pretty much cover those fissures. So the fissures they develop below the cotyledonary node, but we cover them until the unifoliate because soil is soft. After
Mm.
some time they go down. So we cover a little bit up and try to make sure that those fissures were covered. But yeah, I mean it's not feasible overall for growers.
Okay, so maybe Justin could speak a little bit more to the machinery that or the equipment that you used.
Yeah, Gary asked a good question, and we get this a little bit. We don't focus on it too much because such a low percentage would ever use it. But it's a Hinneker hiller. They're built in Minnesota. They're more or less wide sweeps that you have to have the ground tilled. You could do it in no-till. It's really hard to do, but it shoves that soil from the center of the row outward. Buffalo cultivator's another one that could do it. I've had growers who have inter-row cultivators that will speed up. But essentially, as Tolly's did a really good job of describing, it's throwing that soil against the plant up above the first node to really the second node to gain good coverage. And we have a number of studies that have shown it works well. Topography, you have to be really early. Topography is a challenge, and you have to do this around V2, at latest V5 by Pragya Gupta, a student in the lab study, to gain some value from it. So, yeah, if you're looking at, there's previous webinars on it that put quite a bit of focus.
Okay, thanks, Justin and Thales. Yes, I think that's the questions that we have for now. But I'm thinking maybe we could move on to our next presentation. Bob Cook, he's professor and extension entomologist at the University of Minnesota, and he is going to be talking about some of the adult biology, which there's just so many questions. I can't wait to hear Bob give us all the answers. So take it away, Bob.
Yeah, so as Aaron mentioned, I'm going to give some updates on some ongoing research in our lab where we've established a colony for producing soybean gall midge in the laboratory year round. And then from that, we've been able to spin off some of those insects into various different experiments. And I'm not the one actually doing the dirty work, performing those experiments. Want to make sure we acknowledge the folks that are really doing the work in my lab, James Menger, Regina Stackey, and Phelan Anderson. And just kind of a quick outline of some of the topics that we'll be covering here quick. We've got the timing of adult emergence and specifically, how does the emergence of females compare to that of males? And then within the females, we're looking at egg development and egg load meaning the number of eggs per female. And then after those eggs hatch, what is the sex, male or female, of those offspring? And then the longevity of the adults. And you might be asking, why the heck should we care, right? Some of this sounds like kind of nerdy entomological stuff that folks like some of us at the university might be just kind of interested in from the basic science component, but just want to emphasize that integrated pest management is really founded on that understanding of pest biology. So this research is kind of the intelligence gathering upon which we can start developing or improving our integrated pest management programs, right? So it might help us figuring out when we should apply certain tactics or monitoring methods. So before getting into it, I want to just do a quick overview of the soybean gall midge life cycle. The previous presenter touched on it, but at this time of year, soybean gall midge are going to be inside of cocoons as third instar mature larvae, and they're going to be in last year's soybean fields. And then come springtime, those adults are going to emerge and they're going to fly to soybean. Within the soybean fields, those adults are going to be looking for cracks or fissures in the stems where they're going to lay their eggs. The eggs will develop into larvae inside those stems. The larvae will pass through 3 stages that we call instars. And then once they reach that third or mature instar, they're going to drop to the soil to pupate inside of a cocoon. And then they'll start the life cycle over again, with maybe 3 generations per year. And what we're going to focus on here is that adult stage. So the first experiment, again, is looking at the timing of adult emergence. So what we did here is we were able to take the mature larvae out of soybean stems, or the third instar larvae out of soybean stems from our laboratory colony. And then we put them into sand vials so that they could complete their development, forming their cocoon and pupa, and then emerging as adults. And we were checking those vials daily for adults, and then we'd count the number of adults and determine how many were male and how many were female. So I'm going to show those results on this graph here. We're across the bottom on the x-axis. We've got the number of days after vial creation. So after we transferred those larvae into the vials. And then on the y-axis, we've got the cumulative proportional emergence of adults. So that's going to be looking over time. I'll just show it here. For males with the black line and darker gray band, and the females with the dashed line and the lighter gray band. What this pattern shows is, no emergence where it's flat across the bottom at 0, and then the adults start emerging, and then each day you're getting more and more adults emerging. So it's kind of that continual summing up of the adult emergence until all of them have emerged, and then you kind of flatline again at 100% or at a proportion of 1. And we're able to analyze these data and estimate the number of days it took to reach 50% emergence of the adults, or the peak emergence. And for males, that was 10.8 days. For females, it was 11.5 days. And that was a statistically significant difference, indicating that males on average are emerging a little bit before females, about a little bit less than a day before the females. And similar patterns have been shown for some other gall midges, so it's not hugely surprising. And I think some of Justin's field monitoring data has hinted at this pattern as well, with males emerging before females. And then now looking at the development of eggs in those females and how many eggs those females carry. We again used our laboratory colony of the insect, and this time we took cocoons out of those sand vials and placed them into individual microcentrifuge tubes. And then we're able to monitor those tubes closely for adult emergence when those pupae became adults. And then we randomly selected some of those adults at different time periods, ranging from half an hour to 48 hours after adult emergence. And we dissected them to look at their ovaries and then the number of eggs. So here's some pretty cool pictures that Regina was able to get of the soybean gall midge ovaries. So again, soybean gall midge itself is a pretty small insect. So the fact that she was able to successfully dissect out the ovaries from these insects is pretty impressive. On the left, we've got ovaries from adults that were only half an hour old and then progressing to the 48 hours old. And it's a little bit tough to see, but what we see is soon after emergence, the eggs are more round and they're covered with these bright blue spots, which are the what are called nurse cells. And then as the adults develop, those eggs become more elongated and those nurse cells start going away. And that indicates when we've got the mature eggs. And this bright blue coloration is from some special laboratory procedures that Regina used to be able to visualize some of these features. And then now to quantify this egg development, on this graph, we see again that time range across the bottom from half-hour old females to 48-hour old females. On the y-axis, we've got the proportion of those eggs in the females that are mature. And what we see is that half an hour and 12 hours, all of the eggs are immature, so there are no mature eggs in those females. And it takes until about 24 hours until we started seeing some mature eggs. And it was pretty similar, not statistically different at 48 hours. So at 24 and 48 hours, we finally started getting mature eggs, and it was just a little over 50% of those eggs that were mature. So in addition to kind of characterizing the eggs, Regina was also counting the eggs in each female. And what we did in this graph is we measured the tibia of the adults, which is a segment of their legs. And that's been shown to relate to the size of the adult. So we're using the tibia length as kind of an indicator of female size. And then we've got the total number of eggs in that female. And then you'll see some different colored dots and lines that show the age of those females. So I'll walk you through the data. Here's that scatter of dots with those different colored lines. And what we see is that bigger females, again, females with those bigger leg segments, have more eggs, and that the average number of eggs per female is about 177. And then if we look at these letters in the legend there, those statistical differences that we see with the letters indicates that those females that were 24 or 48 hours, the slightly older females, actually had slightly fewer eggs than those younger females. And we think that that's due to either those females kind of resorbing, kind of like within their bodies, like digesting and utilizing the nutrients from those eggs, or those females may have been laying a few eggs in the vials, but we weren't able to recover those eggs or quantify them. But overall, it was a very minor difference over time. Transitioning now into the sex of the offspring. So we looked at, when are those adults emerging? What are they carrying for eggs? And then now what happens when those eggs start hatching? So for this experiment, mated females were placed into cages with individual soybean plants. And then we extracted those mature larvae from the stems, put them into sand vials, and then monitored those vials for adult emergence. And again, in these cages, it was just one single female adult per cage. So we know how many offspring and what sex of offspring were produced by each individual mother. And then we're going to show those data on this graph here, but I'm going to kind of hold your hand through it because it's a little bit complicated. So on the side of the graph, we've got 40 different individual females, individual mothers. And then on the x-axis, we've got the number of offspring from each of those mothers, how many were female or how many were male. So then the bars here again show the number of male or female offspring for each of those mothers. The darker bars are the females, the lighter bars are the males. And what we see is that 22 of those 40 mothers produced only female offspring, highlighted in red here. 16 of those mothers produced only male offspring. And then of those 40, only 2 of the mothers produced offspring of both sexes. And among those 2, mothers where they produced both sexes, the majority of the offspring were females. So again, this is another fairly common phenomenon within gall midges. It's called monogeny, where the females are producing largely just one, individual females are producing offspring of just one sex. And then moving into longevity, so how long do those adults survive? Phelan in my lab set up these vials where we can find individual adult gall midge that were 24 hours old. 3 different treatments shown here where they had moist cotton in the top of the containers where the water could be added to the cotton or not. So the water could be available or not. And then this little additional cage was put over the tip of that cotton to either allow access of those females to that water or to prevent access of those females to the water. And then we set up some of those vials in growth chambers with high humidity at 80% and some at low humidity with 20%. And then we'll see the survivorship curves here where we've got the time on the x-axis across the bottom, and then the percent survival or proportion survival on the y-axis. So for females that were placed in vials with no water available, no liquids available, we see that females in the high humidity environment survived, 50% of them survived out to 2 days, whereas if it was a low humidity environment, they survived only up to 1 day. So they appear to be sensitive to that ambient humidity around them. And then if water was placed into that cotton and they were prevented accessing that moist cotton, their survival increased some, but it increased more when it was in that humid environment. So we had 2 days or 3 days for that time to 50% survival. And then the situation with these blue lines where those adult females actually had access to that water, they weren't prevented from accessing it, 50% of those populations survived up to 5 days. The longest surviving individual is only out to 8 days. So overall, these insects are not very long lived. So we see that they did better with higher humidity and even better when they had access to water. And then we started wondering, why could this be? Are they actually able to drink? And this seems like kind of a no-brainer because we're so used to animals being able to drink, but not all insect adults can. So we tried to test this in our lab by giving females either just plain water or water with food coloring. And what we saw is those females would kind of take on a drinking posture, put their head down to that moist cotton, and it looked like they were drinking. But we were able to confirm that drinking by seeing their abdomens turning blue as they were ingesting that water at the blue food dye. Phelan and Regina dissected open some of those adults and found that their guts or stomachs were blue inside as well. And then kind of the final bit of evidence is that they were excreting blue liquid out their back end. So not only were they taking it in, they were pushing it out the back end as well. So we're pretty confident that soybean gall midge does indeed drink. So just to wrap things up here, the males emerge about a day before females. Females contain about 177 eggs, bigger females with more eggs. Females generally produce offspring of only one sex, and longevity of these adults was greater in higher humidity, and especially when they had access to water. So Erin, I'm not sure how we're doing on time here.
Yeah, that was absolutely awesome, Bob. And you sort of played it low key that just to do all these experiments, you are able to successfully produce an ongoing colony, it's a huge barrier to the research for soybean gall midge the first couple of years. And so now that you've kind of perfected that process, you're able to do all these really awesome experiments. And I thought that was excellent. You answered a lot of questions about biology, egg production. Yeah, that was great. I think that We didn't have any specific questions on this, but there may be as we go along today. So thanks, Bob. That was awesome. And it sounds like you're getting a lot of help from your lab to do that too.
Right.
So thanks everybody in the Cook Lab. Speaking of, next up is Phelan Anderson. He's a graduate student in Bob's lab at the University of Minnesota, and he was part of that adult biology project, but he's also going to share a little bit more about cold hardiness, which was some of the questions that people had. They submitted ahead of time. And so I hope Phelan will be able to answer some of those questions that you had. I see his slides and I think he's queued up. Go ahead, Phelan.
All right, hi everyone, and thanks for that introduction. Yeah, so I guess we can hop right into it, talking about what we found most recently about the cold hardiness of soybean gall midge. All right, so I think we all know why we're here, talking about soybean gall midge, aka Racelia maxima, or as I'll refer to it, SGM. So I think we all know it's a relatively new economic pest of soybean in the Midwestern United States. I'm sure some of you have seen fields that look like this one on the right. It can have pretty significant impacts, especially towards field edges. And if we take a look at a map here, so the first really widespread outbreak where we started to notice soybean gall midge a lot was in 2018. And on the right, we have in orange highlighted the counties where it was detected in that original outbreak, with a backdrop of just soybean producing counties in the region. And then continued surveys throughout the years up to 2025 have found it in new counties every year. And now this is its current known range. So Bob gave a good highlight of the life cycle already, but I'll just remind everybody that the soybean gall midge affects plants by feeding internally. Its larvae gain access to the plant through naturally occurring fissures just above the soil line, which typically start around the V2 to V3 growth stage, and that internal feeding causes a distinctive darkened lesion like this one on the right. If you peel it open, you'll find the larvae feeding inside, with more mature larvae tending to have this kind of bright orange color. But the part of the life cycle that we're especially focused on for overwintering is what happens after they reach maturity. So earlier in the summer, when larvae reach maturity, they're just going to drop out of the plant, go into the soil, make a protective cocoon, and pupate within the soil. Then they will emerge as adults later. But if that process is happening as we're getting closer to winter in the late summer or fall, those mature larvae are going to leave the plant and cocoon in the soil, but they've been getting some kind of environmental cues to tell them that winter is coming soon, and they're going to actually overwinter in the soil within those cocoons. They'll then emerge as adults in the next year's spring. So this is really the life stage that we want to focus on if we want to try to evaluate cold hardiness. So we know where they winter, but what do we know about their actual capacity to survive different cold temperatures? So we've done some previous work on this, and some of that has been presented in the previous webinars. But we know that they do appear to be freeze intolerant, so that means that they die when they freeze. Unlike other insects, some can even survive freezing, and some die before they freeze. Alongside that, we see that they have pretty suppressed freezing points. So rather than freezing around 32 degrees Fahrenheit, they actually freeze around -4 to -14 Fahrenheit. And that's where we see high mortality as well. But there are some caveats there. So we measured this by actually pulling them out of soil that they were cocooned in, sand in the laboratory. I'll talk about that more. But they didn't have this substrate around them or any moisture. And this was also an acute measurement. We watched them get to the temperature we wanted to expose them to, then pulled them out right away to see if they survived. So in order to connect this better to field conditions, we probably want some more realistic conditions. So we probably want to evaluate their cold hardiness when they are buried in substrate and using longer exposure times at relatively warmer temperatures, which is something that we call the lethal time. And to really highlight that, there's some data on the right here. This is from a site in Lamberton, Minnesota. And on the y-axis, we have the coldest temperature that was observed at a 2-inch depth, which is roughly around the range that SGM will be buried over the winter. For a given month on the y-axis, and every dot is a different year's observation of that coldest temperature. But my main point here is if you look at that -4 to -14 range, the soil basically never gets that cold. So we should really look at some warmer temperatures for longer periods of time. And that's the objective of this study, to record the lethal time of buried SGM cocoons. To do this, we went out into the field and collected infested stems along the edge of a soybean field in southwestern Minnesota in late August, brought them back into the lab and dissected out the larvae that were inside and transferred them into vials of sand. So we basically simulated them dropping out of the plant into the soil coming into winter, but done it in the lab. We set up these vials with 10 larvae each and held them at simulated winter conditions, which started out with a month of acclimation at 37.4 Fahrenheit. And then we wanted to pick a colder temperature to transfer some of them out to. So if I show our temperature plot again, the soil temperature from Lamberton, we chose a temperature of 26.6 Fahrenheit. So it's just a bit below the freezing point of water. And it's also a temperature that they might very realistically experience in the winter in the soil. So we split our treatments into these two conditions, and then we pulled out 5 vials from each treatment at each of these time intervals over about 2 weeks to see whether the larvae in them were still alive or not. Let's take a look at our data. So on the y-axis here, we have the proportion that were dead by a given time in days on the x-axis. And first I'll show our warmer treatment. So they did just fine at this temperature. This is generally expected. They had been at this temperature for about a month beforehand already. But our colder conditions, they fared much worse. So we can see that overwintering SGM larvae survive extended periods of time at 37.4, but not 26.6 degrees Fahrenheit. I'm going to just focus on these colder conditions data now. We also were able to calculate the LT50. So this is the amount of time in days that they need to stay at this temperature for half of the population, 50%, to have died. And that's about 6.5 days. And the LT90, 90% mortality here, is about 13 days. So in summary, we knew already that there was this high freezing and mortality following brief exposure to that pretty cold temperature range. But SGM appears to be much less tolerant to longer-term exposure to much warmer temperatures in the soil, where about half the population was killed after 6.5 days at this temperature that's really just a bit below freezing. For future research, we plan to collect more soil temperature data and try to compare this particular data to find out, well, how often out there on the landscape would they actually experience like 6.5 days colder than 26.6 Fahrenheit? We don't know for sure yet, but that's going to be, I think, the next step to really put these numbers into context and try to understand risk of establishment across the region. With that, I'd like to thank everyone who was involved and thank you all for coming, and I am ready for any questions.
All right, thanks, Phelan. I'm wondering if you have a sense if Minnesota has a temperature weather network and for soil temps, and you have an idea of maybe what the 24 or the 25-26 winter was like for you guys?
So that is something we're kind of actively trying to figure out, especially there are wider range networks I'm aware of that are recording soil temperatures at a much deeper depth. Like I think there's a pretty broad 10-inch network, but SGM doesn't really dig down that far. So we're trying to target this like 2-inch depth, relatively shallow. So we're still in the process of trying to track down data for that, and that's stay tuned for future insights, hopefully.
Yeah, we have a weather network at Iowa State, and one of the metrics is a 4-inch soil depth, and they have individual days, and some of those days were in the teens, as far as kind of like you were talking about the extreme kind of moment of cold. But
Hmm.
I don't know if there's a way that we can generate sort of like the not so cold but extended, colder days, a few days in a row to see maybe if there's potential for mortality.
Hmm.
Yeah, I don't know if you're getting those kinds of questions or like Bob and Justin, is it cold enough to kill rootworms, Japanese beetles, and soybean gall midge? But yeah, that was great. We are
Thanks.
going to just change topics for a minute and but in a different way, talk about movement in the soil profile. So we get lots of questions about that too for soybean gall midge. So we're going back to Nebraska. Talia Romez Rodriguez is a graduate student. And let's see if you can bring up your slides. She looks cold. She looks like she's wearing all the layers. Yes.
I'm always cold, yes.
Yeah, so yes, she is going to be talking about soil profile movement. So we can hear you, Dahlia, and your slides are getting queued up here. Yep.
Well, morning, everybody. Thank you for the introduction, Dr. Hudson. Again, my name is Dalia Ramos Rodriguez. Today, I'll be discussing the evaluation of larval movement of soybean gall midge in the soil to see potential insights for research and management. Uh, when it comes to some things that we know is that, well, first of all, some tillage studies have been done in Iowa and Nebraska. From these studies, we have a bit of a inconsistency of results due to variability of overwintering. And let me put my pointer really quickly. If we focus in this line here, we would say, oh, tillage has a great impact with when it comes to adult emergence. But when we combine both, which is this yellow one here, when we combine both fall and spring, we see that although there was a heavy soil disturbance, there's still adult emergence. And because of this gap in knowledge when it comes to larval movement, it's where I'm thinking of focusing myself. uh, some of the things that we do know about movement of larvae in the soil is that after they drop, 95% of larvae stay within that first 0.75 inch from the soil surface. Uh, let's say that we till that area, we could assume that larvae would be moved deeper into that soil profile. And of course, they'd be disturbed. And from here, a question arises, and is, do larvae redistribute themselves within that soil profile? Meaning, will they go up for more advantageous positions, or will they go down? Uh, from here, we first start by collecting infested stems, and our goal is to create a soil profile or to create a disturbance of larvae. And the way that we achieve this is by creating a soil profile and placing them in the middle. This soil profile is achieved by cutting a PVC pipe in half and filling up one segment and placing all the larvae. After we have all the larvae placed, we topped it off, and in total, we created 4 reps. These 4 reps were left undisturbed for around 3 weeks. After this 3 weeks, what we did was divide them into 2 segments, and we would push the soil. And the way, if we go to this picture here, you can see that each layer that we would push down and cut and separate would measure around 1 inch of depth. And why were we doing this? Our goal with this was to see where within that soil profile the larvae were located. And after washing and processing, we got some numbers. Uh, when it comes to distribution, what we saw is that most larvae were found in that bottom tower, specifically from the initial point of placement to that first inch in that bottom section. But when it comes to this top 4 inch, wanted to know movement of the larvae. We wanted to make sure that we weren't. When it comes to movement, we created a no movement zone in which we marked it from 1 inch upwards and 1 inch downwards. And what we saw was that most larvae were concentrated in that inch to the second inch of depth. But we also saw movement throughout that whole segment from the 2 inch to the 6 inches in depth. Uh, Overall, what we saw was that most of the larvae moved downward, and we collected around 86% of them in this tower or in this segment. And the rest were collected in this area with some upward redistribution, with around 14% of larvae collected. Unfortunately, we did not collect cocoons, and this could imply that there's still some movement to that we missed. And some things that I do want to improve on is that I want to simulate this more into more realistic conditions in which the larvae are placed and quickly they're covered. Because we could assume that tillage or aggressive tillage could help, specifically if they're moving downwards after being placed at in a certain layer from the soil profile. With that said, larvae mainly moved downwards within the soil tower, although there was some upward movement observed. These findings suggest that larvae displaced can reposition themselves, indicating that timing and intensity of tillage may influence redistribution to the soil profile. After a bit of mistakes here with the presentation, I appreciate everyone for hearing me. Thanks for the funding from North Central Soybean Research and the Nebraska Soybean Board. And with that said, any questions?
Yeah, thanks, Taya. I think we got the majority of the story. I'm wondering if you or Justin could comment or even speculate if tillage did push eggs push pupa further down into the soil profile, you think they would still be able to complete development and then make it back to the soil surface? What do you think?
Yeah, that would be something that I would love to see later on in different studies that could come from this. But if Dr. McMecken has something better to say, I also will let him talk.
Yeah, we don't have specific data on that. We did have another student do a project where they buried stems at certain depths, and obviously, as we got deeper in the profile, we saw less emergence. I believe Bob's group has a recent paper just 7 days ago that was published where they placed larvae and cocoons at deeper depths too. So he would have some data where they couldn't move around as part of that, and he can comment.
Maybe Bob could give us the short condensed version.
Yes, we use those vials in the laboratory, like I was showing before my presentation, with sand, and then buried pupae in cocoons, or buried cocoons at different depths. And I think pretty much anything buried below 1 centimeter, we didn't have any adults emerging.
Oh, okay.
And then if the deeper they were buried, they were more delayed emerging as adults. And then when we buried larvae, we were getting adult emergence out to 8 centimeters of depth at a very low level. 2 centimeters, still pretty good emergence. 4 and 6, kind of intermediate emergence, but like 8 centimeters was kind of the lowest. Okay. And I'll leave it at that.
Okay. Well, it's interesting to think about just, they physically can't make it to the soil surface in more like field conditions, or would they be like physically crushed, like they don't survive that like physical kind of control. I don't know if you have Bob or Justin have any thoughts about that.
I mean, one other interesting angle is it's not just so the larvae can move some in the soil, but then after that pupa is formed, right? The larvae turn into a pupa, and that pupa, when the adult's almost ready to emerge, it seems like they work their way out of their cocoon and they can kind of shimmy their way up into the soil. And
Okay.
typically, if adults are going to emerge, it's when those pupae work their way up to the soil surface, and then the adults will be emerged. And that was documented previously for other gall midge, including the raspberry cane midge, which is very closely related. And we were seeing that in the experiment we did.
Okay. Okay, great. So thanks, Talia and Justin and Bob on that one. It was a tag team effort. We are going to popcorn back to Minnesota and hear about some biological control and parasitism of soybean gall midge. Sarah has been presenting for a number of these webinars. She must be seeing the finish line, must be very near for you, Sarah, I hope, with graduation in the near future. But we're excited to hear about some of the biological control happening. We had a couple of questions that I think will be addressed with your presentation. So we can see you and I hope we can hear you. Oh, while she's doing that, I can't believe I forgot to talk about this. Hot off the presses is a new pocket size field guide for soybean gall midge. And there are literally boxes, I think, in all of our offices right now that were delivered, paid for by the North Central Soybean Research Program. And so hopefully if you see us at field days or upcoming events, or if you stop by our office, you can get some of these. It's about 40 pages, some of the things that we talked about today, but then more, description, life cycle, all that kind of stuff. So please find us to get some of these for you, your clients, newbies that maybe don't know how to scout for soybean gall midge. If you're sort of on the leading edges, north, south, east, west, grab a copy, find us. We're happy to get those out to you. So
awesome. Thank you, Erin. Let me. Okay, great. Awesome. So I'm going to talk about parasitism of soybean gall midge. And so first, I kind of wanted to give an introduction on what a parasitoid is. So parasitoids are insects that lay eggs inside of a host insect and then feed on the entire host until it emerges as an adult. So our lab has previously identified 2 wasps known to parasitize SGM. First one is Sinopius maximum, and the second one is Sinopius ruficoxum. The first one was found in Minnesota in 2021. That was the first sighting of it, and it was determined to be a new species. And then Sinopius ruficoxum was also found in 2021, but in Nebraska. And there was one previous record of this species before we started seeing it in soybean, and it was found in Canada, actually. From our fieldwork in 2022 through 2024, we assessed fields for parasitism in the heart of SGM's known range. You can see here in orange, we have counties with known SGM. And then in that panel A, we have a black box that shows the range where we collected from various fields in the 4 states that we assessed. And then in panel B, across the 3 years, we sampled 42 fields throughout the field season. On each sample date, we collected 40 random stems on the field edge and dissected out the larvae and preserved them in alcohol for use in molecular methods. And then we screened all of the SGM larvae for wasp DNA. So we used molecular tools to detect the presence or the absence of wasp DNA of either of these species in the SGM larvae. And we did this to see which larvae were parasitized by either of the wasps. So basically, if there was wasp DNA present, that meant that that single SGM larva had been parasitized and would have been killed by this wasp, and a wasp adult would have emerged. Across the 3 years, here are the highest daily percent parasitism rates for both species in each state. We see the highest level for both parasitoids was in Nebraska at 22% for Sinopius maximum and 60% for Sinopius ruficoxum. And we can see just how high this is for Nebraska compared to our other states. Despite the state differences, both Synopia species were detected in all 4 states that we assessed. That leads me to 2 objectives today. The first one is to characterize SGM parasitoids spatially and temporally, and the second is to identify landscape factors that can impact parasitism. So first we'll characterize SGM parasitism spatially and then temporally. Now remember that we found the highest daily percent parasitism in Nebraska compared to our other fields. And so in the most southern fields, maybe there's something going on there. So we decided to look specifically at how latitude impacts parasitism. So here's a map showing the lines of latitude from the most southern fields at 41 to the most northern fields at 45 that we assessed. And then this figure shows latitude on the x-axis, again with south as 41 and north as 45. And on the y-axis, we have proportion of SGM parasitized. And then the color of the lines will show our 2 parasitoid species. Yellow will be for Sinopius maximum and blue for Sinopius ruficoxum. And here are the results. So starting off with Sinopius maximum, parasitism was generally consistent despite latitude. You pretty much just see a yellow horizontal line. It doesn't matter what latitude was, it was pretty low. But if we look at Sinopius ruficoxum in blue, we see something a little bit different, where at the higher fields, or the most northern fields, and the most southern fields, there's actually higher parasitism. So it looks like there may be 2 separate populations of Sinopius ruficoxum, one in the southernmost fields and one in the northernmost fields. And then there's not as much going on in our middle range. Next, we'll look at how parasitism varies temporally across the season. Here's a figure broken out by year in 2022 through 2024. On our x-axis, we have Julian day ranging from 170, which is early June, to 250, which is late August. And on the y-axis, we have our number of parasitized larvae from all of the fields that we assessed. And then our color again of our line will show our species of parasitoids. So here are the results. Now in 2022, there's not enough data, so we're going to skip over that and look at 2023 and 2024. So for Sinopius maximum, which is our yellow line, we see less parasitism happening throughout the season compared to Sinopius ruficoxum. But in 2022, we see that there are kind of these 2 peaks, where one, we just caught the end of it at 180 Julian days, and the other is around 230 Julian days. Then in 2024, it looks like there is only 1 peak at 210 Julian days. And this peak could be equivalent to the peak at 230 in 2023, where it just came earlier in 2024, so maybe there's a year difference. And if that's the case, then the first peak of parasitized larvae in 2024 may have come earlier than we were actually able to capture from our sampling. So maybe it was actually around 150 Julian days, which was before we collected our samples. So maybe we just missed that with our collection. Then for Sinopius ruficoxum in blue in 2023 and 2024, there are 2 distinct peaks, one at about 190 Julian days and the other around 230 to 240 Julian days, and that's pretty consistent between the years. And these parasitism peaks show an alternating pattern between Sinopius maximum and Sinopius ruficoxum, which suggests that the parasitoids are complementary to each other, where each parasitoid fills in a different temporal niche throughout the season, kind of covering the season full of parasitism. Okay, moving on to identifying landscape factors that may impact parasitism. Literature has explored different landscape effects in terms of biological control, specifically parasitism and other systems. And one landscape factor that has been found beneficial to parasitoids is distance to forest edge. And in general, forest has been shown in some studies to provide resources, such as floral resources like nectar, as well as provide alternative hosts that they can lay their eggs in, and provide refuge, so like a hiding spot for the parasitoids in agricultural systems. And so we wanted to look at how distance to forest was impacting parasitism in soybean gall midge. So for each field, I calculated the distance to forest edge by uploading this cropscape data layer, which is this raster layer of different landscape types with different colors you can see here. So yellow is showing corn, that dark green is showing soybean, the gray you can see is an urban area, and then there's a bunch of other colors that are a little bit smaller, harder to see in this photo. And then after I loaded that, I loaded the field locations where I sampled from, and then I used spatial tools to calculate the distance to the forest edge in meters. All right, so this figure on the x-axis, we have distance to forest edge, and then on the y-axis, we have our proportion of SGM parasitized. And then again, our color will show our parasitoid species. So here are the data. Parasitism was higher near the forest edges for both parasitoid species, and there was no difference between the species. So there was a similar trend going on for both of the species. This suggests that the elements of forest habitat actually promotes parasitism. We're not exactly sure what element that is, but this could lead to some future research that could assess what's actually going on in the forest edge and how that's really impacting the parasitoid community. So in summary, Sinopius ruficoxum seems to have two populations in the most northern and the most southern fields. Sinopius maximum was not affected by latitude, and remember it had that horizontal line. The parasitoids seem to complement each other temporally, filling in those different temporal niches. And then lastly, parasitism was higher near the forest edges, suggesting that forest offers some kind of resource or refuge that supports the parasitoids. And with that, I'd like to thank the following, and if there's time, I'll take questions.
Yeah, thanks, Sarah. I sure do get a lot of questions about biological control, and one question I get is, I mean, they're not very big wasps, and so you're not likely to see them in the field, or even if you're sweeping, or would you even know if you were looking at larvae if one was infested or not with a parasitoid?
You wouldn't be able to tell until it starts to pupate.
Okay.
And we have seen some of that in our lab. We actually have some pretty cool photos where you can see the wasps developing inside of the larva.
Okay.
But it was pretty rare for us to find that. So you probably wouldn't end up finding it in the field unless you collect the stems and you're trying to rear out the insects. Yeah.
Okay, but in general, more likely to find parasitism on field edges near more diverse, like forested habitats. Okay.
Yes, that's what it's looking like. Yep.
Great. Okay. That's all of our biocontrol questions, other than the ones that you answered during your presentation. So thanks, Sarah. We are going to go back to Nebraska and hear kind of a mashup of updates about cultural control. These are things that are in the hands of farmers to a certain degree, from Justin McMecken. He's a professor, and you cover a lot of crops, but soybean would be one of those. So he's going to be giving us some updates on controlled tactics for soybean gall midge. So.
All right. Thanks, Aaron, and thanks for doing all the moderating today. You're doing an awesome job, so we really appreciate you. I'll just take the next 8 minutes or so to highlight a few studies on soybean gall midge and where we're headed with these studies. And I think this will be appealing to farmers and those that are working with farmers that have soybean gall midge related issues, because they're low cost tactics to implement. And so to get into this a little bit, we started a series of studies over the last few years that have looked at plant and pre-plant related cultural control tactics that might limit fissure exposure and ultimately infestation / injury and yield. And we looked at 3 different tactics. So I'm going to spice these out over the next couple slides and talk about the direction that we're headed with this particular project. All of this, is that fissure near the soil surface up to cotyledonary node that occurs around the V2 stage that makes soybean susceptible to soybean gall midge. And we'll talk about some germplasm related stuff that George and I and Aaron and Adam are doing later that I'll highlight that again if you have questions on it. So we did a planting depth study. Initially, this is the first study of 3, and we planted soybean at a ½ inch, 1.5 inches, and 2.5 inches. And a big thank you to Julia Pinzone, who was in the lab not this past summer, but the previous summer, and actually did this study at 5 locations. And as many of you know, soybeans typically are planted around 1.5 inches. So we've kind of got variations around that to 2.5 inches. Really simple study design. It's a Latin square design, so it was easy to implement in the field. And the results, I'm not going to waste any time going through a ton of stuff. We did 2 separate collections. So we'll just jump right into the first collection, which was in late June, when we typically see populations really start to build on soybean. And what you see is we reduce or increase planting depth, we've got a significant reduction in larvae at that deeper planting. And a caution to this, there are a couple things that are happening. We've got reduced fissure exposure. Julia was able to document that by measuring the fissure exposure length. We've also got delayed emergence, which we know from another student in my lab, actually graduate now, Natasha Umeziu. Delayed planting can have an impact. But regardless, for a farmer, this is something that could potentially be impacting their ability to infest. Now, we should have left it here, and we should have never came back to these fields, but we did in early August, and we see this, first of all, big increase in populations, which is quite common, late July, early August. Again, another graduate student in the lab has since gone on to other things. Vilma Montenegro documented populations peak late July, early August. That's what you're seeing here. The opposite has happened. Now those late, those deeper plantings have the highest population. And to be honest with you, when we get this late in the season, populations that show up in abundance after the reproductive stages have occurred are less impactful to yield from other work going on in the lab. So really interesting study. We're going to move now to other things related to replanting, but keep this in your mind. So another thing, and already touched on by Talia, is tillage. And Talia's focus, if you can see my pointer, was really in last year's soybean field, talking about that tillage and potential movement in the soil. We're going to move over to the current year soybean field. Got corn stalks out there right now. This being disked up prior to planting, which again, I know if you're a no-till planter, this is not a fun conversation, but it's one of 3 that we're discussing. NCSRP projects. So thank you again to NCSRP for funding so much work that we do on this topic. But we were doing a study actually looking at dense vegetation adjacent to fields that might impact infestation. And we decided to implement hilling in this midway through the project or program to say, hey, how much are we really preventing early season infestation? And to do the hilling effectively, we disked areas in the field. And we got concerned with the disking as we started to set up the study that, hey, we probably need a no-till and a disc only check to look at, with no intent of really looking at much else beyond just limiting the population's infestation for later in the season to look at yield. Turns out that disking alone, at least in this study, did reduce infestation. Now, this is much like the previous study. There's a significant reduction as we move from no-till to that, purple bar where it's just disked and planted into, and then disking and hilling, which we've already talked about, which really is effective because you're covering up the infestation site. If we kind of take a snapshot of those 2 out to what a planter looks like when it goes over the top of that, we've got those press wheels that are pushing down with the disc opener in between. No-till ground, no big push into the soil. Disc ground, we see kind of a divot form in those areas. We did no adjustment between each one of them, so it would sink in a little bit. And we think that this reduction or drop may, obviously it impacted the larval count, but there may be something going on. Either the fissures are being covered up, or rain splash, or microclimates might be impacting that infestation with disking alone. Okay, last one. So we've gone through 2 tactics. We've gone through changing our planting depth. We've gone through disking these fields. Now we're in our field, still a no-till field. So we haven't added disking to this, but now we're actually taking that row cleaner you see on the far left hand of your screen, and we're fixing it in place so it can't move up or down. And that's, you see, we're moving a lot of soil in that first image. And then in the next image, we're letting it float, which is what vast majority of us do, what I always do when I'm planting soybeans. And by floating, we're not really pushing any soil around. Interestingly enough, again, not huge differences, but statistically significant. We still got big populations of both. We've reduced the number of larvae on the fixed row cleaner adjustment. And so, I mean, a neat series of tactics, kind of what I call like poking away at soybean gall midge to limit that critical early season infestation. So to tie all this together, we've got these pre-plant and net plant strategies, and they have the potential. Alone, they each one has potential, but probably not enough to be satisfying for a farmer, although every little bit helps. And the opportunity to combine these strategies together is really where we're headed, and we may have an opportunity, we may have some funding to do that in the coming year, stitching those together and seeing if we do get synergistic effects between those different things. But ultimately, what we're looking at is potential reduced fissure exposure from these or microenvironment changes that are impacting gall midge's choice. And so with that, I think I even did it in 7 minutes. I could take a question or two before we move on to the next one.
Yeah, that was great, Justin. So knowing what we know from Bob's presentation, that midges will live a little bit longer if they take in water. They can drink water. Do you think, I mean, we've done some of the, you've done some of the like edge mitigation where you're mowing or burning, you're trying to reduce like the grassy borders around fields. Would that help with some of the other cultural control that you've been seeing?
Yeah, that edge mowing. So that NCSRP study that we kind of glazed by really quickly, just highlighted that it was part of it, had some interesting results. And of course, Bob was part of that study, or the Minnesota group with Bruce. And so we really appreciate all Bruce's work. It's a challenging study to put in. And we saw variable effects. Nebraska saw effects on occasion where mowing would reduce infestation and lead to marginal yield differences end of season. We're talking like 6 to 8 bushel differences. And
Okay.
previous studies that we've done prior to that stage showing that, yeah, we can save some yield. The challenge we have, Aaron, is that our corn planting date is also a big humid canopy out there. And so the bigger it gets, the more it can kind of overshadow, at least in our opinion, that dense vegetation and also the environmental conditions when this is all happening. So back to Bob's work, if it's hot and dry, it probably has a significant, value or change or may alter. But if it's wet, we may not see much effect because we got all this added moisture in the environment anyway.
Yeah, and earlier you talked about some of the hilling equipment, maybe not part of typical farmer setup, but with the row cleaners, that might be something that is something that they could adjust the fixed or the float. How easy is it to change to a fixed row cleaner?
Yeah, I'll be careful because in saying how easy it is, I have a 2-row planter, so
Okay.
I get made fun of everywhere I go planting adjacent to farmers. It's pretty easy for me to change 2 rows. I know growers have pressure capability depending on the planter, so they can actually push down
Okay.
harder on those. So depends on the planter, but at worst, if you have them, it's going in and just locking them in place. And I wouldn't do this to the whole field. I do this on the opening rounds if I was
Yes.
entertaining the possibility.
Okay, that's what I was wondering. So it's sort of a matter of what equipment you have, maybe like the downforce or the pressure and targeting on edges, maybe
Yeah.
close to soybean the previous season. Okay, great. Those are the main questions we had right now. I think we will move on to something that I know nothing about. Super curious is to hear from Kristen Hendrick Stark. She is also at the University of Nebraska, and she is going to be talking about biowrap. And
Thank you for your patience. And thank you everyone for coming today when you may rather be outside in this nice day. So anyway, hello everyone. And as you heard, my name is Kristen. And today I'm going to be presenting a small proof of concept study that I did last July, where I evaluated a physical barrier approach to managing soybean gall midge. So this study was motivated by the difficult management challenges that we have to control this pest that you are all probably aware of. So I'll start by briefly highlighting some of those challenges. So as you can see here, to begin the process of infestation, the adults first have to lay eggs on the stem fissures at the base. And this places larvae in a very protected location where it's difficult to spray them and other control methods that we normally use. So in addition to the asynchronous development, in addition to the hidden locations that these eggs are in, we also have asynchronous emergence, which also makes it very difficult to spray because they emerge over a long period of time and it's difficult to time when you can spray them. So after hatching, you will see the larvae hatch and then they start feeding inside the stem. And then you'll see this typical blackened outside stem. And when you see this, you know you have a problem. And here's that problem. We peel back the stem and you can see orange larvae. And then here's a nice picture of them inside a petiole. So after feeding, larvae drop to the soil. Here they're coming off the stem into the soil. And then they complete their development and reemerge again. So what would happen if we stop them from getting out of the soil? What if they couldn't get out? So this is the question and our opportunity to think outside the box. So what if we applied a physical barrier at the soil surface to physically block emergence? And this is where the idea for BioWrap came in. I'll briefly explain what BioWrap is before getting into the experiment. So BioWrap is a biodegradable spray mulch that is typically used for weed suppression and soil protection. The name, as you can see here, is an acronym for bioplastics with regenerative agricultural properties. The name, it is made from agricultural byproducts like hay, corn residue, and as you can see here, chicken feathers from poultry production, and then mixed with other ingredients to form a nice liquid that can then be poured on the soil and cures after 24 to 48 hours and becomes a somewhat thin, flexible surface layer. So with that being said, this is the experiment we performed. Stems are collected from the field in July and brought back to the lab. 10 stems were put in each of 9 buckets. 3 buckets contained no BioWrap. And then we evaluated 2 BioWrap treatments at 2 and 4 liters per square meter in each of 3 buckets. So we had a total of 9 buckets. Larvae were then allowed to develop in the sand and monitored for adult emergence for 30 days. So I just want to show quickly here what it looks like pouring out, because a lot of people think it's super thick like syrup, but it actually is very pourable like water. Here we go. So this is what it looked like in the room. We set up the experiment as a Latin square design and kept it under controlled conditions. So here's a summary of the results. We observed a clear difference between untreated buckets and the BioWrap treated buckets. So in the control, we had 30 plus minus 12 adults emerging at an average per bucket, and all of the buckets had adult emergence. And this occurred, all of the adults emerged 12 to 13 days after being put in the bucket. On the other side, in the treated control with 2 liters per meter squared and 4 liters per meter squared, we had no emergence in any of the buckets. So these results indicate that under treated conditions, no adult emergence occurred. So this is exciting, right? But before you get too excited, let me tell you what the application rates are in the field. And some of you have probably already done the math, but that's a lot of BioWrap. So at 2 liters per meter squared, that's about 2,000 gallons per acre. And at the higher rate, it's 4,000 gallons per acre. So for comparison, a typical field application is often closer to 10 to 20 gallons per acre. So this is much higher than what would currently be practical, but it's a starting point for understanding whether this concept works. So our next steps would be to reduce this rate and refine this approach and bring this proof of concept idea to a field application. We were thinking about focusing on field edges where the infestation is often most severe, as well as treating stem fissures where eggs are placed and the process of infestation often begins, and along crop rows in a banded application where you only apply the biowrap close to where you need it. And that also saves a lot of product. So there's still lots of work to be done, but the findings support the potential of this approach as a management tactic. So with that, I would like to acknowledge my funding. Oh, so in conclusion, well, I gave the conclusion. BioWrap prevented adult emergence in laboratory conditions. Targeting the soil stage shows promise, and BioWrap has potential for a new management approach if we can reduce the application rates. So now I would like to acknowledge my funding and all the people that helped me and help my research in all sorts of ways. And thank you all for listening. And I would be happy to answer any questions about this product if anybody has any. Thank you.
Thanks, Kristen. I'm wondering if you could tell us a bit more about how it prevents emergence or how it causes mortality of either larvae, pupae, or adults. Can you tell us a little bit more about BioWrap?
Yes. Originally it was made for, as I said, weed suppression. Yes. So there's a corn protein called cornzine that I showed on my slide, and that actually suppresses root development in some weeds. I don't think that's what it's doing to the gall midge, but I believe if you feel the buckets after they've cured, it's kind of a hard surface, although it's somewhat flexible, but I think they just can't get out. You know, the gall midge adults are quite delicate, and I don't know if they're suffocating under there or they just can't emerge and reach the top, but I think it's just that they can't move and get out of there.
Okay, yeah, when I first saw your description, your title description, I thought for sure it would be targeted to the bases of stems for like preventing larvae from, or preventing egg deposition or larvae from getting in or out of a stem. So I was wondering, do you think it could be more of a targeted application through like drop nozzles? Would you be able to apply it? Or maybe
Yes.
that's what you plan to do.
That's what I'd like to do. I've actually in the greenhouse grown soybean plants and sprayed some to see if it even stays on the stem for a while. I'm going to look at it under the microscope, and you know, that's also one of the problems. How long does it last once you spray it? There could also be a potential for a chemical property of it. Justin always says it stinks so much, and everybody else, whenever I walk around with it in my hands, kind of run the other direction. So maybe they won't like it because the way it smells. But you get used to it. But I think there's a lot of potential, and I have also done just like what would happen if experiments. And last year, when I had some larvae, I sprayed or poured leftover BioWrap on the hoop house soil and placed the gall midge on top of it, and they hated it. They couldn't really move around because it was such a slick surface. So I think the gall midge like a little friction in order to dig down into the soil, because I've also tried putting them on real smooth surfaces or sifted soil, and they also don't like it very much. So,
Okay.
Yeah, there's a lot to do. If we had a few more gall midge in the winter, that would be really nice. Then I could try some of these things.
Yeah, super curious to see the application in the field, maybe targeted sprays and
now I can see
targeted application sites to bring down maybe the cost involved and see more about that. Thanks, Kristen.
Yes, thank you.
Yeah, we have another update from Nebraska talking about some of the germplasm screening, which we've been talking about for many, many years, and multiple states are contributing to this. So I think Justin is going to provide some of the results from screening more elite lines of soybean germplasm. And I think George Graff, who's the breeder on our project from Nebraska, is also a panelist if you have specific questions for him. But I think maybe Justin is going to provide the update today. So yeah, we can see your slides, Justin.
Perfect. Thanks, Aaron.
Yep.
Yeah, George and I talked briefly and felt that the audience here might really want to see the commercial variety testing. So I'm just going to briefly highlight all the work that, Iowa State, South Dakota State, and us have been doing as a timeline, and individuals are welcome to go back to previous webinars to see the progress on that. But it's quite evident that what we've seen in all that work through the years of doing germplasm work transcends to commercial varieties. So there's good opportunity there, and we'll be doing some things in 2026 to continue to further that effort. So George is on here, his lab, our lab, Aaron, your lab, and then Adam Varenhorst in South Dakota as well. And so quickly, just as a one slide to what you can see in previous webinars, is that we've gone through a process. And I always like George given this initial one, which is, and Aaron would appreciate this too, 2020, 2021, these 713 accession lines for maturity groups 1 and 4 should have come with chiropractic care for each of the labs that were doing this. But just a wonderful opportunity to see the diversity out there. We got some great responses, a lot of pressure at some of these sites. And then, through George, narrowed that down to 100 accession lines and have worked with those for a few years. And Minnesota has joined us with their colony to do some control tests through some funding from within Minnesota through their soybean association and group. And then 2026 here, we've got crosses that are being made in elite lines that we'll be continuing to follow up with. What I'm going to focus on for the next couple slides is actually this commercial variety testing, which is a pretty hot topic for a couple different reasons, and I'll do my best to not annoy anybody with that. And I'll just briefly on the next slide here highlight one slide kind of summary of the accession line material here. So of all those accession lines, all were infested. So, some sites, some plots, we may not find them, but we find them in everything. And that's the same when we're going to talk about commercial varieties. And when it comes to fissures, we see different types of fissures, like the one you see here in the image in the center. That's a fairly broad fissure. Some of them were very narrow. This one's infested. It has that black, dark discoloration. But they all have fissures. Sometimes they were really hard to see, but at least at one site or somewhere, we saw fissures on these. And then, that plant injury and larval count, as you see in the graphs here, just between what is susceptible lines and as we narrowed things down, how much we can reduce plant injury, and with the little dots here on the bottom representing larval counts, how much we can reduce larval count. So, if you're really into host plant resistance, there's those components of anti-zoonosis, antibiosis. We don't know any of that yet. But there's some evidence something's going on with these plants, whether it's choice or it's impacting soybean gall midge biology. So this graph will look familiar if you've been to the previous webinar. Just really quick on this one, this is a bunch of XF lines. These are actual lines that are available for commercial purchase. So if you type in one of those lines online, you'll find different companies, available for those. But just look at the continued variation from 500 area under the severity progress curve value, roughly, since we all don't deal with this value. This is like 6 to 8% plants dead. If you move up to around 2,000, it's 40 or so percent plants dead end of the season. But this AUSBC encompasses the whole season. And we're working with industry to try and get this more public. And you can see that in the process, we're initially just kind of highlighting what is the good things in that system. We really like to get to a system where you could see on a 1 to 9 scale what all was available. This is a Davy site from 2024, wide variation in area under the severity progress curve. You put Davy and Talmadge together on this, top 10 at both sites, research sites. There's a lot of shared comparison. That gives us some confidence that what we're looking at is really something that is holding between locations and sites. for the most part. There's a lot of complication with these two. So just a quick from 2024, we're going to spend the next 3 slides on 2025. We're going to start with those XFs again. This is the AUSPC values, a little more pressure at this site than the sites from 2024. We're looking at low end, 12% dead in August, high end, 58% AUSPC encompassing whole season. And I just highlighted the first 4 in this because this was a fairly hard hit site, but you can see that we've got some things even from the previous year that carried forward to the next year. So we got year after year with XFs showing reduced infestation potential. We're going to jump now to enlist because a lot of people are growing enlist lines. We'll spend the next 2 slides looking at some enlist lines. And so in the enlist lines, we've got anywhere from 6% dead to 40% dead. We've got an A in front of a lot of these that you see. That's alloy. That's not necessarily meaning that if you just go out and grab, whatever variety of alloy, you're going to be okay. Alloy also exists in the stuff that we can't see. So just to rate it relative to its occurrence in the top end, that it has also got stuff further down. And I should mention, you're probably wondering, like, where are all these numbers just randomly coming from? I got seed from farmers who were frustrated and wanted to see these tested and have a better way of starting their season. So it's a random, smattering of different things that we got from farmers that we're working with. And then another site here, still alloy lines, at that bottom, and enlist lines across the axis. But you can see pressure, fairly high at this particular location, but between 8% and 43 to 52% dead. If you just look at a one date in August comparison. And a big thanks to Elliot Nell. So I know he's on and listening. Elliot walked all these plots 5-6 times during the season, took all these ratings. The lab, my whole lab went through and took in terms of verifying that larval presence was there. And so a big thanks to him. He put in a lot of hours and effort on this, and certainly we've seen the value from talking with farmers. Where are we going? What do we need? And if those graphs came by too fast, don't worry. I can always pop them back up if you ask the question quickly at the end and we have a moment. Doug is talking about a survey that we're doing, but we really need to get a comparison with them without hilling. I know you all aren't going to use hilling, but this says, if we can take Gall Midge out, how well are these varieties doing? George is doing that with us, Dr. Graff, along with Aaron and others. We're doing that in some of our germplasm work. We're really looking at March is too late to be having these conversations, so we need to get these out October, November, so you have time to select varieties for 2027. And ultimately, with industry, partnering with us on this, like we do for disease ratings, getting a 1 to 9 rating system in that booklet, so you can move varieties around. I've not run across a company yet that doesn't have something good in their system that they couldn't move around. It's just knowing what that is. And we really need to know the value of this to your operation. We're sitting in kind of a nexus of where to go with this. So if you see value, please lean out and let us know. You can drop me an email, what it meant to your operation or anything like that. So summary, germplasm and accession and elite lines show differences in susceptibility. It's pretty impressive. What's available through George's program and what we see in the U.S. germplasm source. We got similar response between commercial soybean varieties, as we look between the two. And this variety selection is really the foundation of these IPM programs. We see Rex in the state, at least I saw them, and often we get enough into conversation if we've tested, we know that variety, didn't hold up as well. And so getting those growers on the right track is a good way to reduce this. So questions or comments before we jump over to Doug?
There were a number of questions about the treatment, well, not treatment threshold. Basically, do we have an economic injury level, economic threshold? And you sort of alluded to that with the area under the curve as far as like seasonal accumulation. But can you say anything more about there really aren't any thresholds because we don't have really great rescue treatments, but can you say anything about like the economics of midges?
Yeah, not too much. I mean, Aaron, the closest we got is with Mitchell Helton in your lab, who I should have given a note to him in the slides, but I mean, that AUSBC came from his work in your lab and some data from us. And that's really the foundation that we have for as we move through the season, what yield losses might look like, right? But to have a threshold, that would be wonderful. The best I could give you is the observation that I've had in the field, and that's not really, it's a type of threshold, but not really, is that early season, if we start to see larvae exceed 20 per plant, and we've got small plants, so we're talking, V5 or prior plants, and excess of 20 larvae, we're looking at plant death coming pretty soon.
Yeah.
And so that's something visually in the field. If you want to estimate the risk that you're sitting in as gall midge emerges and after those 10 days after, that's where we sit. But this is not a pest that we really react to during the season. So we can't really lean in and stop what's happening. And so that's been the challenge.
Yeah, and it sort of reminds me of if people are used to thinking about cumulative aphid days for soybean aphid, it's the 90+ days that they could be feeding and causing injury. So the area under the curve is sort of a seasonal accumulation of larval activity because of multiple generations. And so, yeah, you're right. We really don't have like a treatment threshold or like a rescue treatment option at this time. So we know that they certainly can cause yield losses, plant death, and that's, directly relates to yield, but we don't really have great answers on like how many plants does it take, how many larvae per plant. That's still in development.
Yeah, and I just a brief thing, and then I'll push it over to you and Doug again. It is challenging because when gall midge shows up, we get compensation if it's early enough and plants die fast enough. So sometimes you can get these wild early season results, but if it's a smattering, soybean makes up for it. And so it's a complicated scenario.
Yeah, we have a few more questions coming in, but I did want to leave some time for Doug Golick. He's a professor at the University of Nebraska, and he has, I think, a pitch for you guys. So take it away, Doug.
Yeah, thank you very much. I do have a little pitch here at the end with my stuff. I'm going to talk to you about one of the initiatives we have. I do some work with Dr. McMecken, with Justin, and everybody's least favorite thing. When you say the word survey, most people run. But I did want to share one of the things we're doing that is funded by the Nebraska Soybean Board. Can everybody see that survey? Hopefully you can.
Yep, we can.
This is, I'm going to put up in the chat here in a minute when I finish up the Soybean Gall Midge Network website. It is soybeangallmidge.org, which is, I think, runs out of Dr. McMecken's lab, but I think a number of people contribute to it. It's a great website in itself that has a bunch of information. You can sign up for text with alert messages where outbreaks of soybean gall midge, you get an alert when they first occur or first noticed in the fields in the spring, and other emergent dates. But in the middle of the screen, we do have a link to a survey. The survey is funded by the Nebraska Soybean Board. We've been running the survey for the last 2 years, and we are in our 3rd year. This survey is for soybean producers and anybody that works with soybean producers. And the data that we are interested in, the information we're interested in this survey is your concerns about soybean gall midge and other emerging pests. In Nebraska, that includes Decty stem borer. I know in other states you have that as well. And things like herbicide-resistant weeds. So we have been collecting data on those. Also, your management and preferred management solutions to dealing with those emerging pests. So we've been collecting that the last 2 years. We're on our sort of final round or final year. We may continue to do this in the future. And what this is helping us do with all the faculty in their labs and other people that have been on this call today and the students, you can tell they're all, besides doing the basic research and very applied research, they're all very interested in, because all extension people, very interested in how that affects the actual producers. They're not doing research that isn't eventually going to have an applied aspect. The kind of work that I do on the human side is very much that way as well. We want to hear from you, because if the solutions don't make sense for you and can be used in the field, essentially what's the purpose? And so this survey is helping collect that data, and it also informs them a little bit about what current concerns and trends are. We also work with the Nebraska Soybean Board and others, and we hope that informs them about what your needs are as producers and folks that work with them. So I do want to share a smattering of data. I don't want to pollute this too much because I don't want to bias you if you are going to do this survey, and I really encourage you to do it. We need responses on this. some things we found. Of course, herbicide-resistant weeds are always a concern, and the primary concern over a lot of others. But in the last year or two, soybean gall midge is approaching as near a high of concern as herbicide-resistant weeds for survey respondents. And again, there's some bias in that with people that responded to the survey. You're coming here on this meeting presumably because of soybean gall midge issues, but that is for a lot of people becoming a near as large of a concern. And of course, one of the top management strategies people are using is crop rotation, because the other strategies really that's the one that people are trying. So those are some bits of data. What are we going to do with this when we finish this year up? I presented last year at our Society, Entomological Society of America. We'll be sharing this data out for 3 years and showing some trends so that it'll inform what we do in the future. So with that, I know there's not a lot of time. I want to pass it back, Aaron, and if the people have questions or thoughts or concerns. I will say one other thing before I do that. There is an opportunity if you want to just, I want to talk to people too. So there's, I am giving out $25 random gift card, but if you can also sign up for an interview to talk with me. I'm easy to talk with. And I would like to talk to people, even if you're not a producer, about your concerns or thoughts about this issue. So there's an opportunity to do that as well. Easy Zoom call or phone call. So yeah, nobody likes also doing interviews, but I'd love to talk to you about what you're doing.
Yeah. Great. You said the word gift card and my ears perked up a little bit. So yeah,
a random drawing. I can't afford to like 100 gift cards.
Right. Well, yeah, you gotta play to win, but sounds like a couple different ways to provide feedback. I know that we all take feedback when it comes to generating new research ideas, how people like to get information through extension events. So we appreciate you, Doug. Yeah. I have a couple of questions that didn't get answered as we were going through the last couple hours. So there's a couple of rapid fire questions I'm hoping that people might be able to answer. Bob, when it comes to mating behavior of the adults, do you know how long it takes before like a female comes out of the ground and she mates with a male? And then maybe is she mating once or is she mating a couple times over her short life?
Those are very good questions and remain to be determined.
Okay.
Yep.
Yeah, and some of that you'd have to maybe glean from maybe other well-known midges, but it's a very short lifespan as an adult compared to maybe some other pests. So yeah, it'd be interesting to know that, but generally males are coming out first, as we would guess. Females come out, but we don't know kind of what happens after that.
Right. Yeah. I mean, we even saw, so I showed like over multiple days that males are coming out slightly earlier. We also have some data looking at within a day.
Oh, okay.
And males are emerging a little bit earlier in the day than the females. So they're ready to try to do their thing. They're not very long lived, so I'm assuming they're gonna start the process soon.
And big props to whoever can dissect midge ovaries, because I just can't even imagine the technical finesse it takes to do that. So thanks, Bob.
Yeah, that was Regina.
Yeah, Sarah, someone had asked, and it's a really great question. How did you define a forest for your GIS analysis? You don't typically think of these states as like highly forested. Maybe northern Minnesota is a little bit different, but how did you define a forest for your analysis?
Yeah, so in the cropscape layer that I use, that raster layer, they quantified forest as a 30 by 30 meter like pixel, like plot. So each pixel represented a 30 by 30 meter area of forest. So it wasn't just looking at like 1 or 2 trees. It was a big area. And it also included deciduous forests, conifer. And so there were a couple different forest levels that I included all together to label as forest. But I used the different categories that the cropscape layer gave me to develop what I then considered forest.
Okay, 30 by 30. So it's not just a couple of trees on the edge. So
yes.
Thanks, Sarah.
Yep.
Phelan, we had some follow-up questions regarding your overwintering study. Do you think that soil moisture has an influence on survival? And if so, maybe how much? And then what about freeze-thaw cycles? And this week is a good example. I think we've experienced like almost 100-degree temperature swing, at least in air temperatures. Can you talk about maybe soil water and then maybe freeze thaw cycles?
Yeah. So in general, I would expect that there probably is some kind of an influence. One of the things that we think might be going on for why they seem to die at much warmer temperatures that we haven't actually proven yet, but a plausible explanation is there's something called inoculative freezing, which is where they may have suppressed their freezing point way below 32 Fahrenheit. But if water in the environment that is in contact with them starts to freeze, that can kick off. I see. Can't hear me. Okay. I'll bring this closer. Is now better? Sweet. Okay, so just in brief, I was talking about this thing called inoculative freezing, where although SGM suppresses its freezing point quite a bit below the freezing point of water, um, if it is in contact with water that freezes, that could potentially cause it to freeze at warmer temperatures. Now, we don't know for sure that this is what is going on, but that's kind of an idea of how soil moisture could influence winter survival. We haven't explicitly tested that though. In our case, we were using, well, I haven't tried drying out the sand that we use to figure out like what percent moisture that would end up being, but we have been using a standardized amount, which seems to be kind of somewhere in the middle. It's not really towards saturation yet, but it's not dry. And when it comes to freeze thaw cycles, I think that could certainly be relevant, but we just haven't tested it directly yet.
Yeah, I just think of what's happening in my backyard. I'm seeing little midges, and then I saw like over the weekend some really, really harsh air temperatures, and wondering kind of what's going on below ground, not only for midge, soybean gall midge, but lots of other things that try and overwinter in the soil. So,
Right.
okay, we'll depend on you for more details down the road on overwintering survival. Thanks, Fallon.
Thanks.
So I'm wondering if maybe Bob or Justin or actually anybody can comment on, do we have any information on new insecticides, like either systemic, seed treatments, at plant, foliar? Do any of you guys have any updates on insecticide performance?
Yeah, I don't know if either one of us really wanted to click in and answer that. It's a tough topic. So, and I'd be happy to have Bob in your comments as well, Aaron. I think when we discuss this topic, we see people get results, we see people that don't get results, and it doesn't make a lot of sense why some have success and others. So it may be down to duration of emergence at sites or other things that are going on in those fields. Maybe it's a mix of variety that's smashed in with that when we don't have experimental designs that are really trying to answer that question, but people just spraying. We've seen some effect of that combination product. Something that includes a pyrethroid is typically where we start to see something. I think that the challenge and the caution and the reason we're not discussing it in this webinar is that it's hard to take those fields to yield differences. So we'll see it early in larval count in the field. We'll see them drop off. Once we get further into the season, then the Helton Mitchell score for AUSBC start to climb. We might see subtle differences there. And by the time we get to yield, it's washed. And that's just the duration of adult emergence and length during the season that makes it challenging. We do have ongoing studies. NCSRP is funding a methods application study. We did that last year. There's some hints that drop nozzles or high pressure can increase efficacy, but I think those same long-term seasonal issues exist. And feel free, Aaron or Bob, to push back on my comments.
I second that emotion, Justin. All what you said is what I experienced too. I don't know of any new active ingredients, new formulations, new placement, as far as that people have been asking about. I don't know if you've seen anything in Minnesota, Bob?
Well, just to follow up a little bit. So we've been doing some work in the laboratory looking at toxicity of different insecticides to the adults, right? So exposing them to dried residuals of the insecticides, which is a pretty standard laboratory method. And I'm glad it aligns with what Justin's saying, but the pyrethroids are doing a very good job killing them, and even pyganic, which is an organic pyrethrin, so kind of what the pyrethroids are modeled after. That actually worked very well too. Belay was pretty decent as well. So I don't think it's that if we can expose these insects, I think they'll die. So I'm assuming the challenge is just getting the insecticide to them and for long enough, right? Because the challenge is, the stuff you guys talked about, plus, multiple generations, long periods of emergence of these adults. So I'm optimistic that we have products that can kill them. And then, with hopefully our combined forces, we can figure out the biology and, improve the timing and application methods, right?
Yeah, so it's a matter of contact. We haven't found really a great way for insecticides to contact larvae with the products that we have labeled in soybean. I see some new, more, there's different kinds of group numbers coming out in the world, but not necessarily targeted to field crops. And so that's of interest. I get a lot of questions about some new products coming out that we just haven't looked at yet, and maybe is a possibility.
Yeah. Mentioning the larvae, I know Justin's group was doing some work with toxicity to larvae. The stuff I was mentioning was just focused on the adults here.
Okay.
Yeah, I can brief that over really quickly. That was seed treatment related imidacloprid testing on larvae, and we got a dose response curve. And so yeah, it exists. It's the challenges of even at max labeled rates in the field for a grower perspective on the use of that, which is through Natasha's work. We got similar results. We just got that early season effect and then lost it later.
Is that because, do you think just the concentration of those neonics is petering out?
Yeah,
too soon in the field?
Yeah, and to add a little bit to that, Bob, as we get injury to those stems, I get concerned about the amount of injury in those areas and what insecticide is left there, and how they're feeding within that zone. So not that it may be critical, but it's something that we raise as a question.
Yeah. I think it's going to be ongoing and maybe a complement to some of the other cultural, genetic, and biological control that we heard about today. Chris, go ahead.
One really quick one is that if somebody is really interested in like the all-out approach, there's a couple webinars back. There's a study we did with Syngenta where we did 3 applications back to back, and that's really maxing out this opportunity. And go look at it, because I think it'll make you pause about coming back to these fields repeatedly and just trying to cover the window. We're going to see other pest issues if we keep doing that in fields, and the return wasn't that great. Even when we amp up, I think we're asking for other problems.
And think about the input costs of spraying at least 3 times. The kind of that break even, we really haven't realized that as well. So, Kristen, okay, there was a ton of questions about BioWrap that I think it was a new concept for me. And so if I could summarize, there's just multiple questions of a similar. And one real basic question is, does it break down, and how does it break down? Is it like sun and water or something else?
Yeah, I'm sorry. Earlier, I realized that floating message was me not sharing my screen. So here I am. And I thought you'd never ask. I've been saving this sample for 3 months. This is what BioWrap looks like after you peel it off of soil.
Okay.
Like a little piece of leather, if you can see that. So I have tried those things. Yes, it does break down, and that's the purpose of the product, is to add nutrients back to the soil as it slowly breaks down, protect soil erosion, soil from eroding, and as well as keeping moisture in the soil. So I tried that myself to see if it worked, and got this little sample and put something underneath and slowly poured water on it. And it didn't go right through immediately, but it's sort of like a little sponge. And so there's little micro pores where the rain and or water could go through, and then it keeps it under there for a while.
Okay, so you're still getting infiltration of water.
Yes.
Okay, that was one of the questions, but you don't know exactly how fast it breaks down in field conditions.
Yeah, they have tried all of those things. It's made for weeds, so I have had that sample and been watering it in the hoop house daily, and it was several months, but it slowly gets hot and breaks down and turns into like little paper. So
Yeah,
whatever's left, maybe the whatever the keratin from the chicken feathers is just left there. So it's white and
okay.
um just it really looks like paper after it's broken down and then just falls apart. But
Okay,
it
so it would
lasts
be
quite.
a reapplication. It would be not a one and done. It would be ideally multiple or like once a year, maybe at the most. Yeah.
Yeah, definitely
Okay.
once a year, targeting, when you think it's the most important. I don't think it will totally suppress something, but
Yeah.
if you can reduce the numbers so that you can get ahead of it, I think it has potential for
Yeah.
that.
I would
And
love to see this in a field day. So, yeah, that
yeah,
would,
I poured the leftover, I didn't know what to do with onto my berm at the house. And like 3 weeks later, I said, "Oh, let me check what it," and it got all the mulch. And I ran outside into the garage when I saw the question, but I couldn't find it fast enough. But it formed a hockey puck pretty much on my berm that I could
okay.
pick up. Yes,
That's great. Yeah, I would love to see it in person in the field.
yes.
There was a question that came in before our webinar asking about the potential expanded territory in Minnesota. Can you comment on maybe range expansion?
Like in regards to the new counties that popped up this last year or
Yeah,
so?
I think so. That was all that was on the question.
Yeah, I mean, so now we've got it in Minnesota. If you remember back to the map that Aaron showed, essentially from the border with the Dakotas all the way to the Wisconsin border, particularly coming through the Twin Cities. And that's where we made the majority of the finds, or all of them in the last year or two, were around the Twin Cities area.
Okay.
Some of those were in soybean, but most of those were in sweet clover, which is one of the alternate hosts. And in some places, it seems like it's been almost easier for us to find them in sweet clover than it was in soybean.
Okay.
But yeah, I mean, we're up to, the counties bordering Wisconsin. So
Yeah.
kind of assuming if we or somebody starts looking there, it'll probably pop up there too.
Yeah. That's what I kind of had in my mind as
But
well.
I guess with all that said, still in Minnesota, in soybean, we have to work pretty hard to find it in a lot of fields in a lot of these counties. It's not the levels of, in many areas, it's not the levels of infestation that Justin's seeing in Nebraska.
Yeah.
We have some areas in the western part of the state, extreme western part of the state, that are very heavily infested in some years, but a lot of the fields, it's kind of imperceptible probably to most people. It's been Bruce
Yes.
Potter and his crew were very cued in on finding this pest, who made a lot of those detections.
Yeah. We had also a question that the map that I showed at the very beginning was just a positive detection. It was no indication of severity or number of fields infested or anything beyond just that first detection. And so even though as Justin corrected me, it represents about 19 million acres. We don't have a way surveying effort right now to show how many of those fields are infested, at what level are they infested, and so on. So we don't have that type of detail with our surveillance programs yet. At this point, we're kind of doing like first positive detections and sort of watching the range expand north to south and east to west. So we had just a general question about what was the pressure like in 2025, and I think maybe you sort of summarized it for Minnesota, Bob, but what about Nebraska? I can also comment on Iowa. What do you think, Justin?
Yeah, I mean, Tully's work kind of captured, if you put a susceptible soybean in an area and plant it at the wrong time, how bad things can get. Like, I think he covered that at the start. So that's kind of worst case scenario. But we've had, and his data captures that, south of Lincoln, a dramatic increase in pressure where I'm having the conversations I had in 2020 with farmers where they were like, down there, like, we heard it when it came out, we ignored it. We kind of knew it was maybe in our county or not. And now it's like, my operation is significantly impacted. If there's not a decent solution, I may move away from soybean in a significant way because I can't tolerate the loss. So
Yeah.
that who knows if that'll continue into 2026 here, but it was apparent in 2025.
Yeah, I would say somewhat similar. The areas that have been infested in the past, I think it'd be easy to go to those farms and find soybean gall midge. But the severity was pretty variable. Although I did see and hear about some fields where it was significant plant loss, still mostly at the edge, but the plant loss is sort of creeping in more than I had seen in the past. And so some fields, I think just like data planting, maybe a susceptible variety, the conditions were great and they did very well. The midges did very well. And so I think there's a lot of frustration about that as well, because it sort of flexes up and down. But I think if you're looking, you would be able to find them in most counties, like on the western side of the state. So
Yeah,
I'm not sure if that was a good summary.
that was an excellent summary.
Yeah.
And just to briefly add to that, we had other studies going on in other areas. One was on TAPS, which is this technology, approach that growers can do in teams to test. And one of the varieties happened to be a variety we tested. And in that study where that group used it, they lost $157 per acre because of that variety. It
Yeah.
stood out amongst the others, just a significant drop.
Yeah.
And so I mean, I think a lot of what we're seeing, Aaron, and Bob, it doesn't have as much pressure up there, except for the odd year, is really about variety conversations where growers have moved away from things and things got better, and some are just bumping into stuff on occasion.
Yeah, actually, some of the newest detections that we had on the eastern side was a farmer thought that he had a lightning strike, because there was with surveillance, like drone surveillance, noticed a couple like dark patches. And actually, like ground truthing, moving to those areas is when he was like, oh, this is not like a lightning strike, or like a patch of dead plants or like deer damage. Like he was able to take a picture and show me, and I was like, yep. I mean, it was pretty obvious. So I think the word is getting out about, some of the visual symptoms to look at too for those new counties. And I think all of us would take that type of information, a screenshot or something like that, and send it to us to keep that map as accurate as we can.
Aaron and
Bob,
Justin, I'm not sure how we're doing a time here.
we're in overtime, but there's still a few people hanging on.
And Phyllis just caught me and said that probably some of them are looking for a QR code. So
they're
we're holding them
waiting. Oh, I'm
hostage.
sorry. They're waiting
So-
for that QR code. I apologize. I got excited about some of the deep dives. So yes, Phyllis, if you'd be kind enough to share that QR code for people, we can continue on with just a few more questions. I apologize. Yeah, we're in overtime.
Not everyone is as excited about bugs.
Maybe not everyone's as excited as me. Speaking of